we use pollen extract, encapsulated. However, pollen is not very soluble. That's why we have to use proliposomal technique. But here we make a modification. So without using any tris hydrochlorur or hydrochloric acid or heating technique, we obtain good results, and we increase the bioavailability up to 4 times, as you can also see on the orange ones, proliposome results. Another study is adding sour cherry phenolics into yogurt. We also saw that even in the storage period, the encapsulated liposomal encapsulation also keep our bioactive compounds. As a conclusion, in the near future, I think encapsulation techniques, especially liposomal ones, are expected to have more application area in food industry due to their being biocompatible and providing increase in bioavailability. But we have to solve, we have to overcome some degradation and stability problems. And also is, I hope it will be possible to, to use them in large production systems. These are some examples for my encapsulation studies. I would like to thank you all for listening me. Thank you very much.
Thank you very much, Professor. So is there any question? Maybe we can take one question. Okay, so it looks like there is no query. So we'll move to the next presentation. So next presentation is by Professor Ivan Salomon. So Professor Salomon, please, you can start when you are ready. Maybe your microphone is muted.
Professor Ivan. Professor Ivan is not here now.
Yes, it's okay.
Thank you. Thank you very much.
Yes, I am ready. Yes, thank you very much for my possibility to give my presentation. Yes, that I would like give my presentation. Yes, that I would like to thank to my possibility to give the presentation to the MESMAP-6. I think that this is very nice meeting now, and we obtain a lot of useful information from different parts of the medicinal, aromatic, and spicy plants, and of course the techniques for its proceedings. My presentation has the name "The Active Substances Accumulations of Chamomile Varieties Under the Growing Conditions in Chusa, Peru." This is some cooperation with the company, the Luge. I will explain it, and it's very interesting for comparing the possibility to grow the chamomile in the another soil and climate conditions. That the first is very important.
Professor, can I request you to make it fully screen?
I try this. Well,
yeah, thank you.
That is connections with your. Yes, that this is my presentation. I going to the introduction. Yes, that my favorite plants is the chamomile, Matricaria recutita. You have some information about the history. Is that the plants is going back from the time of Egyptian pharaohs. Of course, is very useful and using in our phytotherapy at present. The composition about 220 chemical constituents have been identified. Is that you have the whole spectrum of these components. The main substantial part of drug effects are the essential oil, and in general, that the contents of essential oil in the flowers is from 0.24 to 1.9% of fresh or dry plant tissue. Of course, the composition of essential oil is very important, that the main constituent and most active constituents are the alpha-bisabol and chamazulin. This is the whole spectrum of secondary metabolites, chemical structure that I mentioned about the alpha-bisabolol, yes, about the alpha-bisabolol oxide A, about the chamazulin, and this is the flavonoids. I will mention about the determination of apigenins. That is very important, the determination and identification point that this is the flower that is without, it's empty as the flower. This is typical for the chamomile, German chamomile matricaria recutita. 20 years ago was determined 4 different chemotypes. It's very important to us because if you are looking for the morphological characteristics of the plants, that this is there are the same, but the chemical composition is different, and you can see that was identified in general 4 different chemotypes. Of course, the composition of essential oil in regards to these different chemotypes is very different and very various. That is very important to looking for the plants with high contents of alpha-bisabolol and chamazulin. In regards to these questions that we have the breeding in Slovakia, we bred several the varieties. Last varieties was bred at my university, University of Prešov, Slovakia, in 2015. Currently, this variety obtains the certificate by the Community Plant Variety Office in Angers in France. This is the Europe Union certificate. If you obtain the determination by GC or LC, is that we have about 67% of bisabolol, 10 or 14% of chamazulin, very low contents of bisabolol oxide. The contents of essential oil in these varieties from the 0.65 to 0.85. I mentioned about the flavonoids, that is very important for the isolation into extract the apigenin 7 glycosides that we obtain, and the results about 0.33% of apigenins as in general of this apigenin 7 glycoside. In regards to our cooperation with very big company Lush, Fresh Made Cosmetics, you have some of the history, and this is the last product from last year, the chamomile flower buzz bomb. Yes, that we obtained a very good cooperation. Last has been the created Fresh Made Cosmetic for humans all over the world. Yes, produced the soaps, shampoos, shower gels, lotion, and another the products, yes, for the everyday using. And from Lush we obtain the questions that it is possibility to introduce our chamomile, our variety chamomile lianca into the Peruvian soil climatic conditions. and we starting very good cooperations with this company. And in regards to possibility to use another the varieties, you have mentioned that this was the variety Lianka, variety Peruvian, cultivar Bodegold, this is the German variety, and Zloty Land, this is the Polish variety, that it was possible to compare the contents of essential oil, the composition of essential oil, yes, composition of the extracts, yes, and in these conditions, that is in conditions, the Peruvian conditions, the Andean mountains, and the altitude is from the 3,500 meters to 800 meters. That is very interesting, the altitude. Yes, this is very high mountains. The first, the information is in table 2, that you can see the weight of the 100 chamomile dry flowers heats. Yes, that you can see that the very high grams have the variety Zlotilan and the variety Bodegold, but it's normal because this is the tetraploid varieties. It means that these flowers are bigger than diploid variety, the variety Lianka and Peruvian cultivar, but to the very good weight, yes, the yields for the chamomile is in variety Lianka. You have the information where is it. This area, this is the capital, Lima. In Peru, this is about 550 kilometers from a distance between the Lima and this area. This is the distinct San Jeremio, where was cultivated this for different cultivars or varieties. This is the information about the equipments that we are using at my university for the GCMS analysis and HPLC, that analysis, and you can see the chromatograms from the Lianka and Peruvian cultivar as for the illustration. Very important is contents of essential oil. It was too very interesting. You can see the contents of essential oil in percentage and quantitative essential oil contents per 1 gram dry raw flour material. That you can see that the highest contents of essential oil was determined in our variety Lianka, 0.55%, it means this is 5.5 milligrams per gram. And another, it's lower. Another varieties and cultivar is lower. Is interesting because usually the tetraploid varieties have higher contents of essential oil and quantity of essential oil than the diploid varieties. Of course, very important is information about the composition of essential oil. That here is the totally good results to our variety because we have composition about 60% of the bisabolol contents and about 13% of chamazulin contents, very low bisabolol oxide contents. Yes, in compared to another varieties and cultivars. And if you calculate it per milligram to 1 gram of the dry flowers, that you have very big differences because you must calculate it with the contents of essential oil and contents of the different components. That, for example, for the diploid variety Lianka is 3.3 yes, milligrams per gram, and the better from another varieties, Zlotilan or Peruvian, this is 1.2 milligrams per gram of bisabolol. The same is in, for example, in chamazulen and bisabolol oxide. You can see bisabolol oxide B is the highest in the variety, the Bodegold. It means that our variety has the best the composition in regards to phytotherapeutical activity and is possible to use for the cosmetic preparations. The very interesting was to the determination the apigenins and apigenin 7 glycosides, that the results is similar. In general, this contents of flavonoids is depend on the soil climatic conditions. It means the micro soil climatic conditions. There are not very big differences, and of course, it depends that is the lingular and the tubular flowers. That the lingular flowers is higher contents or than the tubular flowers. And you can see that the results to our variety is very good, that it's possible to use for the extraction, yes, for the dry extracts or liquid extracts, alcohol liquid extracts. This is some of the pictures from the cultivation in the Peruvian conditions. Yes, that this is the farmers who are cultivating each year the chamomile. This is the populations of our variety Janka. This is the hand picking of collecting by the scams. Yes, that for example the scams were supported from the Slovakia, and it is the finish the yield. Yes, dry material which the company using for the distillation and uptime for this the company products. For the comparison with the Slovakian cultivations, you have these pictures. Yes, that we have the full mechanized, the collecting the chamomile flowers. Yes, this is the quality of the collections. That is more percentage, about 70% clear flowers. Yes, and this is after the sorting process. Yes, and cutting process. And this is the final products. This is the dry chamomile flowers. That is very nice. Of course, in Europe is very popular chamomile. We have a lot of the teas, yes, which are used, selling, which are selling in our shops, yes, and the people prefer very good this teas, yes, and another the products too. That this is my conclusion. Breeding efforts were successful in the developing the chamomile diploid variety Janka of better oil composition. The tetraploid varieties Lotilan, Bodegol, and Peruvian diploid cultivar in Andean mountains. The altitude more than 3,500 meters under sea level. The total oil contents and the alpha bisabol contents of oil were highest than other varieties from Poland and Germany. The variety Janka has plant population which are characterized by good yield, resistance to the stress, bigger flower heads, and sufficient amount of essential oil of appropriate chemical composition. The chamomile variety Janka and its large-scale cultivation, extraction, and distillation is very forward raw material to today's pharmaceutical industry. I am one from the leader for the chamomile research, and I think that in regards to my cooperation with the people around the world, we can give this like the conclusion. Nowadays, chamomile research and development is reaching the very high level, and it's carried out from different aspects in various parts of our globe. The present ranges of varieties, cultivation, and proceedings of chamomile with high quality of the essential oil have the possibility to fulfill the demand for this raw material all over the world. This is some of my memories from the first symposium of Hamomal research development and using. It was in 2006, and this is the memories times from this first Hamomal symposium. I hope that sometimes in the future we have the second Hamomal symposium, and of course that you will invite it to this the scientific event. Thank you very much for your attention.
Thank you very much, Professor Solomon, for a very interesting presentation. So here the floor is open for any questions or queries.
I have. Dear Ivan, thank you very much for your kind presentation. When we will start the adaptation for chamomile in Turkey, in Kilis?
But I don't know that we mentioned about this and talk many times, and I don't have any offer to my
So next year,
to my stay in Kilis.
Next year we should start.
It's okay. It's normal. I told you now we sowed the chamomile in Slovakia. Yes, in last week I have the half hectare at this time for the sowing of the chamomile. That we preparing very good the seeds. Yes, seed material, because this is very important in the starting, and we will start in the Kilis. Yes, that you
prepare for next year.
Yes, for it is going to preparing for the next year, because it's very high demand for the Lianka variety, and I don't have the possibility to give a lot of the production at this time. Understand? Because
small field.
You must prepare this seedling materials, and next distribute it. This is important to us. But this year we have very good yield, because it was more cloudy, we have the rain. Yes, this year that the yield was very good at this time. That now we are preparing the products, yes, and export to the abroad by the camions at this time. But we have the trouble with COVID.
But my company also will import from your product.
Yes, that's okay.
Okay.
We will discuss about it personally.
Okay.
You will come here and we will discuss about it.
Okay. Thank you.
Welcome.
Thank you, Professor Solomon and Professor Najim. So next we'll move to Professor Srinivasa Rao Mentreddy. So please, sir, when you are ready, you can start.
I'd like to thank Nazim Sekroğlu and his team for inviting me, as well as a wonderful a global virtual conference, speakers all over the world, and very good topics. I'm so impressed. I'm glad I'm attending this. And let me put this on full screen. Okay. I'll be talking about the application of it. This is it's an emerging technology that's finding a lot of applications in agriculture. It is right there where we are just starting to see how to use this technology for a wide range of agriculture applications. So I'll be speaking a little bit about it within the next 15 minutes. So we tried to look at it from the viewpoint of promoting early sprouting and increased plant growth of brand growth, and also yield of turmeric. Turmeric is a big project for us. We are screening like 60 different varieties from Vietnam mainly. My team here, Lam Dong, is from Vietnam. He's doing a PhD. And then Dr. Xu, he's a professor of aerospace engineering. And he does all the technology part of creating the plasma and helping us treat the plant material. And Dr. Gott, he just finished his PhD He was with Dr. Shu as a graduate student, and he was also working with us, mentoring our students. This picture here is of the turmeric plot we have here in a research experiment station. I'll put a lot of different flowers of turmeric. It's a beautiful plant and very aromatic. So what is this non-thermal cold or low temperature plasma? This is plasma is like a fourth state of matter. It's pretty universal. If you see this, we have solar plasmas, where plasmas in the North Pole, South Pole, those beautiful lights, northern lights, they call them. They're all plasma phenomena. And the high pressure, high temperature plasmas used in industrial applications, whereas cold plasma or low temperature plasma at the room temperature is used in agriculture, particularly soft biomatter, plant materials. If you use any high thermal plasma, it will kill the plant, it will burn the plant up. Therefore, we use either atmospheric pressure or low temperature plasma. So these are actually weakly ionized gas of or ambient air, like air, regular air, of positive, negative ions, electrons, excited neutral atoms, free radicals, molecules in the ground in excited states, and UV photons. And some of these fluorescent tubelets also, they have plasma application there. So plasmas, there are different types of plasma. We have plasma jets, corona plasmas, created from noble gases like argon or helium. And these gases are split by passing electrical energy through the gas in a controlled manner. And what it does is it speciates the gas. It creates like a lot of oxygen, and then OH, and also nitrogen, and these different reactive species. Here are the pictures of some of this. This is a jet plasma we have created here using helium. And then same jet plasmas, we put 4 jet plasmas in this one we made with 3D printing. And we created this small contraption, so we get like a uniform output of plasma. And this one here is with argon gas, and this is a microwave plasma. So there are different types of plasmas using different types of gases and power settings. And these are all the various applications, man-made and natural plasma applications, and manufacturing, particularly semiconductor industry, uses a lot of plasma. Etching is all done by plasma, space propulsion, fusion energy, lighting, all this. And of course, in the nature, we get plasmas due to electrical passing of electricity through these ions, different gases in the atmosphere. Okay, now these are some of the agriculture applications out there. And new applications are coming out because the science is just started growing, and it's still at the laboratory stage. There's not much commercialization because there are a lot of limitations to production of plasma. So nitrogen fertilizer is something you're looking for, where you activate water, and then that water has activated nitrogen species, which will provide nitrate fertilizer liquid. And then also bacterial infections. Plasma tends to kill bacteria, either it inactivates or kills it. And then we have also reduced pesticide residual effects are also being degraded, they're helping in foods without pesticide residues, mycotoxins, pasteurization, sterilization, particularly poultry industry, meat industry, where they spend millions of dollars for sterilization of the foods. Packaging industry also is looking for this food packaging to avoid bacterial infections. all this. So there are a lot of applications in agriculture, but they're all in laboratory stage at this time. So the way we create plasma is, this is the pulse source, electrical source here, and then run it through this. We run the gas through one of these chambers here. As the gas goes in, it reacts with the electricity, and the plasma jet comes out at this end. Okay, this is the plasma jet. We put it through this brush-like item, and we treat turmeric. So this helps us because this is a few millimeters, and you need a lot of jets to um, treat a large volume of plant material. So we came up with this kind of a instrument that will allow us to do like a brush. This is only about a few millimeters from the source. Okay, so turmeric is a very popular crop here. It is gaining a lot of popularity because there was a paper published stating that consumption of turmeric, particularly the curcuminoids, they repair the neuron joints in Alzheimer patients, by patients with Alzheimer's disease. So that clicked a lot of interest here. So industry is going berserk with a lot of products out there. US imports $36 million worth of turmeric. About 90% comes from India, and the rest comes from other small countries. So there is a lot of interest among farmers to grow this crop to cater to the local industry, domestic industry. But then we don't have the varieties, number one. Number two, we don't have the technology to grow. And also there's problem with this season itself. Turmeric needs certain months of good hot season to accumulate enough of those bioactive compounds, curcuminoids. But in this country, we have a short season because of the frost. We get frost late in April, then again in early November, we get frost. So the season is limited to 7-8 months. And that is causing some problem. We get immature rhizomes because a lot of farmers would like to save 25% of the harvested rhizomes for planting the following season, so they don't have to keep buying material. And but if then they have a large proportion of immature rhizomes, then they don't sprout it when they plant them in the field, and as a result they have very poor establishments. Okay, so our study was to assess the effects of low temperature plasma or atmospheric pressure plasma on sprouting, plant growth, yield of turmeric varieties which are known for slow or lack of sprouting. Actually, the way it started was that my grad student bought some rhizomes, and I said, Doc, they've been sitting for 3-4 weeks. They're not sprouting. What should I do about this? So I just casually said, go to University of Alabama, Huntsville, Dr. Gabe Shu's lab, and treat with low temperature plasma and plant again. We will see what happens. So he did a treatment and a non-treatment lots, and then treated that with plasma, put them. Within 2-3 weeks, they all sprouted, the ones that were treated, whereas the untreated did not. So that sparked our interest in this work. So later we did some replicated trial. We first looked at these 5 varieties. These are Vietnamese varieties number 12, 18, 23, 24, and 44. And then we exposed that to helium gas, low temperature pulse jet plasma for about 90 seconds. After treating, we put them in seedling trays and put them in the greenhouse. We used a soilless potting mix and watered the plants as and when needed. Used a randomized complete block design. Okay. This is Dr. Gabe Shu's lab. They have gas over here on the back of this Almira, and it comes out here. This is the machine where the electricity is passed and the plasma is generated. So first time around, we made this using 3D printing. We created this ring-like plasma dispensing structure, and we kind of skewered it like barbecuing the turmeric. put that here. This is Lam Dong, the grad student. This is Ryan Gott. Then later we came with this kind of piece, treating whole turmeric. And then we planted them in the seedling trays. And if you see these treated ones sprouted much earlier. This back row here, they're untreated, same variety VN12. So the treatment did work, and they also grew a little better than the untreated. Okay. So the observations were to check the number of days to sprouting, also record plant height, and if there are any abnormalities, because plasma has the ability to scorch the material. Okay. So we found very good results here. These are the varieties VN12, 18, and 44. The treated plants were growing taller and better looking than the untreated plants in these parts. So we did some metrics on those to see at the days to sprouting, 100% sprouting, and we found that each variety, there was a varietal difference. For example, VN4, they treated, they sprouted within 15 days, whereas VN18 took about 35 days, even with treatment. The untreatment took much longer, about 40 days here. And these are probably early sprouting varieties, but when you treat with plasma, the sprouting is reduced by another 7-8 days. So we found there is a variation among the varieties too, to their response to the plasma. Their variation range anything from 18% to about 93% among these different varieties. Let me see if I can move this out of the way. Okay. So there is an intervarietal variation for the response, and also we found that the plasma certainly improves sprouting. Okay. So then we looked at the plant height. The solid lines here indicate plasma treated, and the broken lines here indicate untreated. So definitely right from the beginning, there is a gap between these two. These sprouted ones are taller and they grow faster than the untreated. All these varieties have showed the same trends. Okay. Okay, those are the results I just not discussed. So then we came with another experiment where we looked at immature and mature rhizomes. That was the idea. We wanted to see if we harvest the rhizomes very early, like in 6 months of planting, what happens. So we selected uniform sized rhizomes from both lots. These were harvested in 6 months, harvest in 7 months. And we treated different times of exposure. No treatment control, 60 seconds, 90 seconds, and 120 seconds. Okay. And then after treatment, we planted in the pots and put them in the greenhouse. So the untreated 66-month-old rhizomes, the untreated never sprouted. They just rotted in the pots. But we had at 60, 90, and 120 seconds treatments, they sprouted very well. 90 seconds, the plants were bigger compared to 60 or 120. Even 7-month-old, the control also sprouted in this case because they were mature rhizomes. But then if you look at these here, 60, 90, and 120, they sprouted much earlier and also grew very fast. So you can see the differences in their plant heights. Okay. So these are the measurements. They are days to sprouting here. 6-month-old, the control never sprouted. 60 seconds, they sprouted in 49 days, whereas 90 seconds sprouted in 28 days, and 120 seconds about 42 days. So there is a difference there. We find 90 seconds seem to be optimal time of exposure. Same thing with 90 with the 7-month-old mature rhizomes. The control took much longer, 49 days to sprout, whereas the treated ones took much less, 60 seconds, about 41 days. This one, 90 seconds, only 21 days compared to these two. Plant height showed the same trend. 90 seconds, they grew much taller and better looking. And than the control, there are no plants at all, 60 and 120. Same trends in mature rhizomes. We find that 90 seconds was optimal. So we then later we transplanted these plants into the field. This is all organic production system. We add only organic manures like chicken litter, vermicompost, cow dung, cow manure, and so on. And these are on raised beds with plastic. There's a drip tape underneath for putting irrigation. And again, in the field, we saw the same trend. 6-month-old control, no plants, 60 seconds, plants here. The little shorter, but 90 seconds, the plants are pretty tall and more leaves and better looking plant compared to 120 seconds. 7-month-old here we find control also. We had control rhizomes, so they did grow. Here the differences between these treatments was not really very apparent, but when we did 4 replication means, we found that 90 seconds had a higher, was sprouted earlier and also taller plants compared to 60 or the 120-second treatments. This is the means of those plant heights. If you see here, this is the number of days after planting, and this is the plant height. And you see this is 0 control, there's nothing there. But then we have yellow 60 seconds, red is for 90 seconds, and cyanide blue is 120. So in all these cases, as the plants grew over time, we found that this 90-second treatment plants remain tall. Okay, this is 6-month-old rhizomes. Whereas in 7-month-old, you don't see that kind of differences. As the plant grows over time, the differences are not really very high. So if we do it, we haven't done statistics on this, but if we do, most likely these won't have any significant differences. But here in the immature rhizomes, definitely there's a significant difference between the 90 seconds and the 60 and 120 seconds. A very big significant difference there. Then we harvested the plants. Yeah, 1 minute. Okay.
2 minutes.
We are almost at the end. Final harvest, again, 90 seconds, double the yield than the control. And here we see that we have even 6 months, probably in 6 months, there is nothing in control, but look at the 7-month-old, it's almost double the yield. Same thing with 6-month-old compared to 60 seconds. 90 seconds was much higher, almost double. But then 120 seconds was just a little better than 60 seconds. So in conclusion, we think that 90 seconds exposure to helium atmospheric plasma is optimal. And low temperature plasma certainly helps in not only sprouting early, also plant growth and yield. Now we are preparing the samples for looking at the curcuminoid content. So we'll see what kind of effects low temperature plasma had on that. Thank you very much. I appreciate your patience in listening to my presentation. Open to any questions.
Thank you very much, Professor. So I think we can take one question. So is there any questions or queries?
Yeah, if
Yes,
I may,
please.
I'm surprised that the plants react to plasma, because where in evolution would they encounter plasma to develop a reaction to? can it be that they are simply reacting to the light that is emitted during the plasma treatment? Because it's known that in plant physiology that plants react to, say, red light and far red light in different ways. Or do you know, is there a mechanism on how plasma does what it does?
Okay, what we're finding is that we are looking for a postdoc to look at the mode of action, how this is happening. It's not exactly the light emission that's happening because of the excitation of those molecules. But I think what's happening is the creation of active species, reactive oxygen species and reactive oxygen nitrogen species. They seem to trigger some kind of enzymatic reactions in this soft matter. One study we did where we exposed pumpkin seeds to plasma and then looked at the seed structure under a microscope, and we found that the treated pumpkin seed had more holes on it. The pores were much more compared to the untreated. So we believe the plasma may have some effect in creating the holes, maybe just a physical effect that is improving the imbibition of water, because that is the first step in seed germination. The treated seeds tend to take more water because of the more pores and kick out the enzymatic reactions and germinate much faster than the untreated. In the case of rhizomes, we believe there may be some kind of reaction with the enzymes because these are active species, oxygen and nitrogen, and I don't know, because this is atmospheric plasma, that there is a lot of other gases in the atmospheric air, and that may be creating some kind of reactions. There are some studies in Japan where they looked at some cell level reactions occurring in the case of plasma treated. Yeah.
Thank you very much.
You're welcome.
Yeah. Thank you, Professor Srinivasa. So now we'll move to the next speaker. So next speaker is Professor Monica Janu from University of Medicine and Pharmacy. So Professor Monica, please.
Thank you. Dear colleagues, dear Professor Shekeroglu, dear Professor Sevcijevic, I am very, very glad to meet you here today. Unfortunately, we were commonly meet in Turkey, but in these days, I'm very happy to see you all healthy and in a very good shape. So thank you for this invitation. Thank you for the opportunity. And I want to ask you to be very flexible. And I want to pass the word to one of my very young and promising researcher in my lab, my youngest colleague, Flavia Burlek, who will present our research for today. Thank you. And I wish you all only the best, a good health, and see you in very short time, hopefully. Thank you. Please, Flavia.
Thank you, Professor. Esteemed professors and researchers, thank you for the opportunity that you have given me to manage to present and deliver the presentation regarding a species that is very dear to me, namely Tagetes erecta. I will try to share the screen now. Sorry. Unfortunately, I have some small issues. Is it okay right now?
Yes, it's there.
Great, thank you very much. So the current presentation will be about investigating the chemical profile, antioxidant activity, and cytotoxicity assessment of Tagetes erecta fractions, which was done in collaboration with the Institute of Soil Science and Plant Cultivation from Poland. I will start my presentation by presenting some information regarding Tagetes erecta, a plant that is well known to, I guess, everyone here. Tagetes erecta is one of the most popular plants of the Asteraceae family, and it is native to Central America, especially Mexico and Guatemala. It was introduced in Europe in the 16th century, and it was brought from Mexico. And it is nowadays cultivated as an ornamental plant, as well as for its high content in carotenoids, which have various uses in the food and pharmaceutical sectors. The species, as you can see, has many synonyms and can be found under different common names, such as American, Aztec, Mexican, or saffron marigold. Its Spanish and Portuguese common names, I'm not sure if I will tell them right, Flor de Muerto and Cravo de Defuntos, refer to the common practice found in Central America of using an aqueous extract from the plant for washing bodies after death, given its pleasant smell, as well as to the fact that in that area, this plant can be often found in the cemeteries. Tagetes erecta is an annual herbaceous plant with yellow or orange flowers that are grouped in solitary inflorescences. And given its high content in lutein, which has various applications, the plant was mostly investigated for its carotenoidic content. Nonetheless, its volatile oil has also been studied, and it is used in the food and the cosmetic industries. However, other classes of compounds found in the plant, such as polyphenolic acids or flavonoids, could present important biological actions such as antioxidant, anti-proliferative, and anti-diabetic. Therefore, we decided to investigate its potential use as a resource of such constituents with biological action. The plant was cultivated in ecological conditions in the northeastern part of Romania in 2017. The inflorescences were ground using a commercial blender. 10 grams of the plant material was weighed, and afterwards 200 milliliters of methanol was added. The extraction was conducted using a magnetic stirrer for 3 hours at room temperature. Afterwards, the extract was filtered through filter paper, and the solvent was evaporated to dryness using a rotary evaporator. The obtained extract was stored at 4 degrees Celsius until further use for preservation reasons. The crude methanol extract was purified with the help of various chromatographic methods. Firstly, actually, the extract was subjected to solid phase extraction using a preconditioned reverse phase C18 column. Followed by the removal of compounds with high polarity, which were eluted with methanol 1% and methanol 50%, while a phenolic rich fraction was eluted with a solution containing 85% methanol and 0.1% formic acid. Afterwards, the 85% methanol fraction was further purified using a Cefadex LH20 column and diluted with 100% methanol. For this part, an HPLC Gilson system was used, which was equipped with an evaporative light scattering detector. And as a result of this separation, 4 representative fractions were collected. The duration of the method was of 1,000 minutes, and the collection of fractions started after 50 minutes, just in order to allow the compounds to pass through the column. The flow rate was established at 2.5 milliliters per minute, and generally the peaks of interest appeared in the first 600 minutes after injection. As you can see, the chromatogram for for the 85% methanol fraction can be seen on the slide. As it can be seen, two peaks appeared. However, due to their wideness and high level intensity, we decided to separate these two peaks into two, given the fact that it could be that under these peaks more compounds were present. So it was a good thing that we did so, because we managed to obtain 4 fractions, out of which the last one was of high purity and was then subjected to other direct analysis without any further purifications. Regarding the LCMS analysis, the methanolic extract was subjected to high-resolution liquid chromatography mass spectrometry analysis. The separations were carried out using a thermo-scientific ultimate chromatographic system and a C18 column. The separation of the compounds of interest was achieved using concave-shaped gradient from 5% to 60% of phase B, which was acetylnitrile containing 0.1 formic acid, and phase A, 0.1 formic acid in distilled water. Over 25 minutes. The flow rate, as it can be seen, it was of 0.55 milliliters per minute. And the column effluent passed through the flow cell of photodiode array detector, recording absorbances in the 200-600 nanometers wavelength range. And the flow splitter was then used to divert the column effluent between the QTOF MS and the charged aerosol detector, which were connected in parallel. The mass spectrometric analysis were carried out in both positive and negative ion mode with electrospray ionization. And data acquisition and processing were performed using Bruker Data Analysis 4.3 software. The chromatograms can be seen on the slide. So we have 4 chromatograms. The first one is for the negative mode ionization. The second one corresponds to the CAD detector, while the third one is for positive mode, and the last one is for the ultraviolet visible detector. We have managed to identify over 50 compounds in the initial methanolic extract, some of which were amino acids such as phenylalanine and tryptophan, polyphenolic acids such as gallic, quinic, syringic, and ellagic acid derivatives, as well as monoacyl and diacyl chlorogenic acids. We have also managed to identify some flavonoids, either as glycosides or as glycans. So as glycosides, quercetatidine, quercetin, camphorol, and patuletin glycosides, and as glycans, axilarin, isoramnetin, patuletin, and quercetin were identified. These are some of the compounds that we have managed to identify in the methanolic extract. I have pointed out quercetatidine because we have managed to separate it in quite a high amount in one of our fractions, our last fraction actually. And besides that, we can also say that the obtained results are in accordance with the data found in literature, given the fact that for these species, compounds such as quercetatidine and camphorol, syringic, and ellagic acids have already been previously identified in different parts of the plant. The 4 fractions collected after preparative chromatography technique was applied were monitored taking into account their chemical composition through LCMS analysis. Some of these secondary metabolites found in the initial extract were identified in the obtained fractions, as well as one can easily foresee. Fraction 1 contains mostly syringic acid and some of its derivatives, such as syringic acid dihydroxy-3-methoxybenzoic acid hexoside, while fraction 2 contains different syringic acid derivatives, such as disyringic acid hexoside. Fraction 3 contains mostly digaloyl hexoside, ellagic acid hexoside, and quercetagetin hexoside, while fortunately for us, fraction 4 contained over 85% quercetagetin, which is actually a glycoside of quercetagetin, namely quercetagetin 7O glycoside. Given its high purity, as I've previously mentioned, this last fraction allowed further NMR analysis through which the structure was confirmed. Regarding the biological assays, the antioxidant activity of the initial extract and of the fraction containing mostly quercetagetin was evaluated through two different mechanisms. So we had first the iron chelation mechanisms, and the second one was the inhibition of 15-lipoxygenase. The capacity of the investigated extract to chelate ferrous ions was determined according to a method described by Venditti et al. With slight modifications. Briefly, the ferrous ion forms with ferrozine a pink complex with a maximum absorbance at 562 nanometers. The presence of a chelating agent in the reaction medium, however, decreases the concentration of the formed complex and consequently the absorbance of this complex. For each extract, the half maximal effective concentration was determined, and it was expressed as milligrams per milliliter final solution. And quercetin, as it can be seen, was used as positive control. The total extract had the lowest half maximal effective concentration, which was even lower than that of quercetin, which was used as positive control, as I previously said. This can be correlated to the presence of different polyphenols, as well as to its high content in carotenoids, which can also contribute to the antioxidant activity through this specific mechanism. The total extract was more efficient than the fraction in chelating iron ions, which can be explained by the higher amount of polyphenols, of different polyphenols found in the initial extract that can form stable complexes through their many hydroxyl groups. The inhibition of 15-lipoxygenase was determined using the Malterud method with some modifications. Briefly, lipoxygenase buffer solution was treated with the extract that was dissolved in dimethyl sulfoxide in various concentrations and kept at room temperature for 10 minutes. Afterwards, linoleic acid buffer solution was added. The absorbance was then measured at 234 nanometers for 120 seconds. For each sample, again, the half maximal effective concentration was determined and expressed as micrograms per milliliter final solution. The results obtained in this analysis are different from the ones obtained in the previous analysis. So we can see that in this part, the fraction seems more effective than the total extract. This is which basically proves the importance of separation of compounds in order to obtain a better activity. Regarding the cytotoxicity assessment, we have used the MTT assay, which is a very popular colorimetric assay developed especially for cell viability screening. This assay basically measures the reduction of yellow MTT to a purple formazan product by mitochondrial dehydrogenases, and its absorbance is afterwards measured spectrophotometrically. When cells die, they lose their ability to convert MTT into formazan. Thus, color formation can serve as a marker of viable cells. The cytotoxicity assessment was performed by evaluation of cell viability in the presence of the investigated samples at 24, 48, and 72 hours of incubation. The absorbance of the obtained solution was then measured at 570 nanometers. For the total extract, a slight tendency of decrease is visible, decrease in cell viability, when increasing the concentration and the contact time, which could indicate a cytotoxic potential at higher concentrations. However, the analyzed fraction which contains mostly quercetagetrine, did not show any signs of cytotoxicity, given the fact that it maintained its the cell viability at over 80% of the control. So in conclusion, the present study focused on a phytochemical HR-LC-MS analysis of a methanolic extract obtained from Tagetes erecta inflorescences and separation of compounds using solid phase extraction and Cefadex LH20 chromatography. We have obtained 4 representative fractions that were further chemically characterized by LC-MS analysis. The last fraction, which presented a high purity and contained as major compound quercetagetrine, was later tested for potential antioxidant and cytotoxic activities, generally showing better activity than the initial extract, which basically gives us an idea about the importance of a separation of compounds from plant extracts. The obtained results basically prove that separations are very important in the quest for substances with enhanced biological activity and lower cytotoxicity than that of the initial extracts. Thank you very much for your attention.
Thank you very much, Flavia, for your presentation about the interesting plant species, Tagetes erecta, and its biological properties. Do you have some questions to this lecture, to this more information? Thank you very much.
Thank you very much, Professor.
For your presentation, and I wish you good and a lot of success and health. It is very important in this time. Yes, that next speaker, I don't know, that in regards to the program, I haven't seen nobody. Nazim? I know that the Alban Ibraliu had presentation. Morning.
No, finished.
No, finish. Alban in the morning. Now, the next speaker is Salvatore Labella.
Now Salvatore, Dr.
Okay.
Salvatore will give speech.
Okay.
Salvatore Labella, yes.
Yes,
I haven't seen. I see, yes, I would like to introduce the speaker from Italy, the Professor Salvatore Labella from the Department of Agriculture, Food and Forest Sciences, University of Palermo. The title of presentation will be Agronomical and Chemical Characterization of some species of aromatic and medicinal plants from Sicily, Italy. Please. Professor.
Okay, thank you.
Yes.
Okay, thank you. Good afternoon, everyone, and welcome to my presentation. Before my lecture, I would like to thank Professor Nazim for invitation. I work at the University of Palermo. Here with my research team, we work about biodiversity of aromatic and medicinal plant as rosemary, oregano, thyme, and myrtle. Today, I will talk you about oregano, that is an aromatic species cultivated in Sicily. In Sicily, we have mainly Oreganum vulgare subspecies Hirtum. This subspecies is qualitatively and quantitatively rich in essential oil, and for this reason could be commercially interesting. The quality and quantity of essential oil can vary due to genetic factors such as the age, the vegetative state, and the part of plant, and to environment factors such as the climate and soil. They can produce, therefore, biochemical difference in plants of the same species grouped in different environments. Cultivate plants of oregano, which include the selection of local biotypes with the best bioagronomical and phytochemical characteristics, would all farmers to obtain a homogeneous and high-quality product. The rational and disciplined use of a specific chemotype can be interesting in order to provide the food and the cosmetic pharmaceutical industries with a standardized and high-quality product. The aim of this research to provide a systematic analysis of the phenotypic variability of oregano plants within a large population of biotypes collected from various sites in Sicily, focusing on biomorphological characteristics, production, and qualitative chemical evaluation, in order to identify, choose biotypes best suited for the local development. Initial research included the collection of wild plant of oregano subspecies from different areas of Sicily. To identify suitable collection sites, a literature search and several field trips were carried out. The oregano plants were collected from each site during the flowering stage. The total of 57 oregano biotypes collected was considered representative of the Sicilian oregano population. The main geographical, topographical, and ecological characteristics of the sampling sites were catalogued. All biotypes were classified as Oreganum vulgaris subspecies Hirtum, and the following characteristics were recorded: plant 8 in centimeters, 8 of flower stratum in centimeter, total number of branches and stem per plant, fresh and dry weight of the plant, dry weight of inflorescence, leaves and stems in gram. Extraction essential oil on sample of 3 gram dry biomass by hydrodistillation, and was obtained essential oil. Determination on essential oil, identification of the volatile components, antioxidant activity by foliage culture, UV radiation induced peroxidation in liposomal membranes. Research on wild oregano population carried out over a number of sites in Sicily led to the collection of 57 biotypes. These were found mainly in hilly and mountainous areas, and on sloping land there was mostly limestone rich in rock patches and with low production potential. Table 1 shows location and altitude of the collection sites of the 57 Sicilian oregano samples. Average altitude was 601 meters and in a range between from 67 from Messina to 906 to Palermo. Table other two show morphological and biomass production after a cluster analysis. The first agronomic cluster contained the largest number of samples, 38, all of which demonstrate relatively good bioproduction characteristics. Biotypes belonging to the second cluster, 5 samples, were characterized by high productivity, making them of particular interest for biomass production purposes. The third cluster, containing 13 biotypes, showed the lowest value and each agronomic characteristic examined. The highest biomass production level were found in the fourth cluster, represented by a single sample, sample number 15. The table number 3 shows here the number of components of essential oil were found to range between 1 and 8 and 6%, with an average value of 4%. In fact, only one sample, sample 29, showed a yield below 2%. 20 samples produced yield between 2 and 4%, whereas 30 samples, 59% of the 51 samples investigated, produced the yield of from 4 to 6.6%. This figure number 1 shows relationship between altitude and biomass production. The most productive group, group number 2, contained biotypes collected at altitudes ranging between 210 and 698 meters, whereas the less productive group, group number 3, included collected at higher altitude 500 and to 962 meters. The first group, characterized by intermediate production values, consisted of biotypes found along at the entry altitudinal range of the study, 6,700 and 1,000 meters. These results show that the biomass production of the biotypes tended to improve as we moved from the high altitudes to lower zones. Table 4 shows components identified in the 51 samples. We found 5 classes. Monoterpene hydrocarbons with 29 and 77%, and main compounds are alpha-terpinene, paracinene, and gamma-terpinene. Next, compounds oxygenate monoterpene with 70 and 63%. Third compound, sequiciterpene hydrocarbons with 2 and 9 and 9.7%. With main components are beta-cariophyllene, beta-bisabolene, and oxygenated sesquiterpene with 1%. And finally, other components 0.04 and 1.08% of the percent, and the main constituent is thymol with 10.52 and 54.42%. Cluster analysis carried out on the compositional data produced the dendrogram shown in Fig 2, which allowed the division of the 51 samples into 6 groups, following an in-depth analysis of the qualitative and quantitative composition of the above-mentioned cis group, it's clear that this division is substantially based on the different quantitative relationship of the same components, namely thymol, gamma-terpinene, paracimene, alpha-terminene, beta-myrcene, carbapenem-methyl ether, thymol-methyl ether, and beta-caryophyllene. Thymol was found to be the main components in 42 of the 51 sample, group 1, group 2, group 3, group 4, and the last 2 groups of the cluster analysis, group 5 and 6, comprising a total of 9 samples, were characterized by the presence of gamma-terpinene as the main oil component, with considerable agar content in group 5. Table 7 shows antioxidant activity. Essential oil from aromatic plant are known to have antioxidant property. These offer the possibility of using these oils as preservative for food and cosmetic. Essential oil can also act as pro-oxidant by means of their volatile constituents, which turn into such. However, the biological effects of essential oil seem to be the result of a synergism between all constituents, as the activity of the main components is modulated by the other minor compounds, which may play a role in defining the membrane lipophilic and hydrophilic fixation and penetration. In this study, we measured the protective capacity of the oregano oils against oxidative damage in chemical assay by Follin-Checouleau methods. The difference between the results obtained in these 2 tests may be explained by taking into account not only the difference between the experimental chemical system, but also the complexity of the vegetable matrix tested in our study. This matrix can modulate the effectiveness towards free radical formed in T2 experimental environments, as well as towards the viability and activity compounds which they contain. The prevalence of thymol group 1 and or the presence of discrete amounts of carbachol group 3 and 4 may be the factor characterizing the oil with the best antioxidant radical scaring efficiency, whilst the prevalence of gamma-terpinene group 5 and 6, and the presence of a significant concentration of sabinene and transsabinene hydride. Group 2 appeared to be the factor related to low degree and antioxidant power. In conclusion, the results of this study confirm once again that the wild Sicilian oregano, Origanum vulgare subspecies hirtum, as regards its essential oil profile, is a thymolo chemiotype species, a characteristics which distinguishes Sicilian oreganum from the same species growing in Greece, characterized conversely by carbachol. The morphological and production parameters evaluated in this study showed large similarities not only amongst biotypes collected from geographically close areas, but also amongst biotypes found in areas distant from one another. More specifically, the largest number of samples, 38, which compose the first cluster of morphological and production characters, represent the most common Sicilian wild oregano and with good productive parameters as well as good essential oil yield, an excellent basis for local development of these species in Sicily. These positive aspects are confirmed by an increasing interest in the cultivation of these species, which included the wild samples analyzed in this study. In the center of Sicily, particularly in the Agrigento and Caltanissetta area, over 1,000 hectares have been cultivated with oregano, an area which is increasing and which yield higher profit compared to other crops. Many thanks for your attention.
Thank you very much, Professor Labella, for your information about the biodiversity of the oregano in the Sicily. It's very interesting presentation, and I think that it's very useful to the producers of the oregano crops in general in Italy, namely, because it's a lot of using the spicy for the food. Do you have some question to the professor? Please.
I was wondering, how do you see the future of the oregano collection and trade? Would people make blends so that you always have the same kind of taste and smell of oregano? Or do you see kind of local variations where you have different types of oregano which are sold like specialty wines or specialty coffee with the origin as a trademark?
Thank you for question. Oregano is a species multitasking for in Italia pizza and for essential oil, for aromatization oil, olive oil, and frequently utilization is the oregano for salad, but now haven't the specialist similarly wine or other food.
Okay, thank you.
The thymol has very good antimicrobial properties.
Yes.
That is very important for the using for the sprays on the another properties. Yes, that we have some research about the comparison the different type and species with the oil production, and the oregano is very best for the microbial properties. Yes. Thank you very much.
You're welcome.
Well, I'm waiting for another next speaker.
Normally, next speaker from Sharafettin Jama from France, but another colleague, Zakia Bobeiche, should give the lecture, but I cannot see her in the participant. There is green pen, but I don't know if she is here or not.
That we have two lectures. Yes, that is the Professor Dr. Bilia Bauer, yes, from Macedonia.
Biliana tried to enter the program. If we can wait a few minutes, if she can give lecture. Otherwise, we will request from her tomorrow. But now, in 2 minutes, we want to give. And in this period, I want to give some information. So we want to kindly give information about International Cosmetic Congress in Turkey. We are in the scientific board, and cosmetic is very, very important, you know. And last 3 years, our team organized 3 international cosmetic congress, and this year also it will be organized as online international cosmetic congress. If you or your colleagues want to participate, they can visit cosmeticcongress.com and send their abstract and full papers. And also in this congress, there are the companies, many companies from Turkey and abroad. If there is any participant who has his or her products, he can present or she can present her products in this congress as well. This will be also this year online. Normally, they organize in Antalya for 3 times, and this year they will organize in December. And now there is no coming. Biljana, I think, cannot enter. Now I'm closing the session. I'd like to thank you, all the chairs and all the invited speakers. They honored us and they shared their valuable information with us. We started Far East and the Far West in America today, from Japan to America. Tomorrow, I think we will start from, Biljana came. Last minute, Biljana came, if she can connect. Tomorrow also we have participants from Far East and Far West from Peru. We have participants from Peru also tomorrow. We mentioned that we have around more than 20 countries participants. I think Biljana, can you hear me?
Jam, I don't know. I haven't seen.
Biljana, her name is here, but Biljana from Macedonia, and she is also working medicinal plants. Today she has a presentation on Euplants regia. Some preparations obtained from the Euplants regia in folk and traditional medicine in Macedonia, but I think she has trouble with the computer. So we don't want to wait more. All the participants today it's very tight for all of us, and we would like to thank you for your kind participation. And if you are available today, we are waiting your participation as audience, and we will have 2 salons, A and B, and tomorrow morning 9:20 a.m. Istanbul time, we will start with 2 salons, Salon A and Salon B. We have just shared the links, all the emails. If you want to participate, if you cannot enter, please contact us. We will share with you our password. Also, during the day, we share all the session via YouTube channel of MESMAP. Also, you can watch again after the meeting from our YouTube channel. Thank you very much for your kind participation. See you tomorrow. Otherwise, see you soon in Turkey or around the world, anywhere. Sometime we can meet with Ivan. Sometimes he say, "Oh, Nazim," in German. Sometimes he say, "Oh, hello, Nazim." We were in
the world.
and another countries. World is very small, and if you fly more, you can contact easily each other. Take care of you. Thank you very much again.
Just 1 second, Nazim, I'm thinking, we would like to propose for having a combined conference, our American Council for Medicinally Active Plants and this MESMAP we can have in some other country. It's convenient for people from the Mediterranean region and also from the Americas to be able to attend. So I'm going to share with you the flyer for the 2021 conference. It's going to be at Rutgers University in New Jersey in June. And I would appreciate if you can share the flyer with all the members attending MESMAP-6.
MESMAP-7.
Sure, thank you.
MESMAP-7 and ACMAP Congress.
Yeah, ACMAP.
Yeah, joint Congress.
Yeah, let's plan for that. Right now we are this 21 we are having in New Jersey and 2022 in Puerto Rico.
Here and we can fly and hope to see you and with you on that. We keep in touch and we will discuss more.
Yeah, we'll plan for 2023 or 2024.
23 is
joint. Yeah.
23 is maybe guaranteed. I hope.
Okay.
I hope.
All right.
And this 23 is very important for Turkish Republic, the 100 year anniversary of the Republic of Turkey. It's very important to us. We can congratulate together.
All right.
Thank you. Good night. Have a nice day. Good evening. And take care of yourself. See you tomorrow. Thank you again.
Thank you very much.